This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 42, for Friday, July 24th, 2009. Hello, everyone. It's Vincent Racaniello, and it's time for another chat about this week's stories in virology. And I have another great crew with me today, starting with Dick Despommier.
Hello,
Welcome,
Vince.
Dick.
Glad to be here.
Here in my office in New York City. And Skyping in from Western Massachusetts is Alan Dove.
Yes, always a pleasure to join you.
Welcome back. And we have a new guest today over the telephone lines, Delthia Ricks, who is a science writer, a health and science writer for Newsday, and the author of a book called 100 Questions and Answers about Influenza. Welcome to TWiV, Delthia.
Well, thank you very much, Dr. Racaniello, and thank you for inviting me.
Well, pleasure. We have a weekly conversation about viruses, and we thought it would be fun to have you join us, and you can say anything you please. We typically have a list of stories that we go over. These are news stories of the past week, and we try and explain them to our audience in a scientific way. And we have usually a lot of reader emails, which we get to, and then our science picks of the week. So our first interesting story is one that we actually didn't get to last week, which I thought was curious. It's a paper published in PLOS Pathogens about a new kind of influenza vaccine. This is a trivalent virus-like particle vaccine. Okay. Now, this is a very interesting concept because we all know that the current influenza vaccines are viruses that are grown typically in eggs, but now also possibly in cell culture, and these are inactivated chemically. Now, a virus-like particle vaccine is very different. It's not infectious. And what they've done here, remember everyone that the influenza virus is an enveloped virus. It's composed of a number of viral proteins, and there are two glycoproteins in the envelope, the hemagglutinin and the neuraminidase, which are antigenically very important. And then there's another protein just below the lipid membrane called the M protein, which gives the virus structure. And it turns out if you synthesize the HA, the NA, and the M protein in cells, you will produce particles that look like viruses. Hence, they're called virus-like particles. And what they did in this paper is they synthesized these particles in insect cells because it's easy to engineer insect cells to make diverse proteins. And these particles are produced, they purify them, and they injected them into mice and ferrets. And what they show is that they make a trivalent vaccine. So the typical yearly vaccine that we get against influenza is a trivalent vaccine. Last year, it had an H3N2, an H1N1, and influenza B strain in the mix. So they took those three viruses, they made virus-like particles for all three, they mixed them together, injected ferrets and mice, and they got a good immune response. And then when they challenged the animals with influenza, they were protected. So the first step, it looks good in the lab. You can make these virus-like particles. You don't have to have eggs, so anyone with egg allergies, it's not a problem anymore. And I think this is probably one of the vaccines of the future.
Wow.
It's not infectious, no eggs are involved, and it's relatively straightforward to make.
I have a quick...
Go ahead.
I'm sorry.
Delthia, go ahead.
Oh, I just had a quick question. Is it a lot faster to make these vaccines than through the usual egg method?
Yeah. I think it's probably about the same. You do not have to make what the so-called high-yielding egg recombinant. So every year you pick a strain and then you reassort it with a strain that grows well in eggs. And so that step would be cut out. And this year, in fact, with the 2009 H1N1 strain, that's been a problem because apparently the yields in eggs have been lower than usual. And so that's a problem because we need a lot of doses of this vaccine for the fall. And so this system will bypass that. But I
think getting away from eggs would probably streamline some other aspects of the production. I mean, one of the big problems is getting enough eggs. They have to ramp up egg production in advance of the vaccine production, and there's a whole cycle to that.
Right.
And then downstream, of course, you have the whole purification from the eggs, and then people who have egg allergies have problems when they receive the vaccine.
Right. Yeah.
Does anybody know whether or not influenza virus is vertically transmitted in birds?
Dick, what do you mean by vertically transmitted?
You mean from parent to child through the egg?
Yeah. Just a question, because I know that we had a discussion about vertical transmission in insects.
I suspect, I have not heard it, but I don't want to give an answer without looking it up.
Maybe one of our listeners knows.
I'm sure they will tell us. Anyway, I thought this is a nice example of a good way to make a new vaccine.
Absolutely.
These are very immunogenic. Apparently, some companies are working on this already, and it seems relatively safe and straightforward. So I look forward to tracking this development. So we'll post a link to that paper. This is a group in Pennsylvania who did this work. And that, of course, introduces virus-like particles, which I don't know we've talked about yet on TWiV. The second story is another flu story. Last week we had fish flu.
Right.
And this week we have dog flu. Did you know there was dog flu?
I did not.
Yeah, well,
I did. I've been writing about dog flu.
Really?
There apparently has been an ongoing epidemic, a low-level epidemic among dogs across the country. I think the first evidence of it occurred in 2005, and it took a while to identify the virus. But as I understand it now, small dogs seem to be more vulnerable to the flu than larger dogs, and I don't know the explanation why that.
Well, it's afraid of the larger dogs.
I presume that next year we'll have one flu over the cuckoo's nest.
Oh, boy. Yeah, Delthia, we have resident jokesters here on TWiV,
So I noticed.
as you've heard, Dick and Alan.
It's how we keep the lab workers away.
Yeah, you're absolutely right. It did emerge 2004. And what I thought was it. So this is not a human strain. It's a dog strain. It's an H3N8.
Right.
Okay? It originated in horses.
Right. I guess I did know this, but I had forgotten it, because the big list of viruses in the influenza group, when they show them on these big CDC website pages, shows all kinds of animals in all directions, and very few of them actually just infect humans.
Yeah.
There's a whole family of flu viruses out there, and obviously this is one of them.
So this was originally a horse flu, which has been known to be in horses for 40 years.
Right.
Now, do we have any taxonomic data on when it might have made the jump from horses to dogs?
Yeah, about 2004.
Really?
So in 2004, cases of an unknown respiratory illness in dogs, and it was initially in racing greyhounds in Florida.
Uh-huh.
So I guess they're all kept together, and that facilitates the spread. And they showed that it was the H3N8 virus, and apparently it was tracked back to an equine origin, and now it's adapted well to dogs, and it's spreading across the country.
What's the effect of it in dogs?
Cough, runny nose, and fever.
It sounds like a familiar story.
And
what about fatalities?
It's pretty mild, apparently. About 80% of dogs have mild form of the disease. But they do, I suppose, with the racing dogs, they immunize. Now there's a new vaccine. That's where I caught this story. There's a new vaccine has just been approved by the USDA. It's made by Shearing, I believe. And I suppose that racing dogs are one of the targets, but it's also for your dog because it could be serious infection.
Sure.
And so it's spreading throughout the U.S. because most dogs are not immune to it. And it's transmitted just as flu is from dog to dog. So they say if your dog has symptoms, influenza-like symptoms, not to bring it anywhere where there are other dogs. But you could also get the virus and transmit it. You're not infected, but you could carry it as a fomite, for example, and transmit it to another dog.
Right.
So I thought it was very interesting, and just another example of flu infecting many species. And here we have a vaccine for dogs. So, and in fact, at the CDC page, they have it called dog flu. So,
well, now it'll be interesting to see if there are. I mean, we don't know what happened in the past. There could very well be circulating strains of flu, right, in dogs.
We do know what happened in the past. When it first occurred in Florida in the greyhounds, 20,000 racing greyhounds died, affecting dogs at 20 tracks in Florida. So that's what brought it to the attention of CDC and veterinary authorities. It was a very, very impressive outbreak. And as they began to investigate, they found that it was affecting dogs in kennels and in grooming centers. And that's how they put the epidemiological picture together. And that was, 5 years ago. So
right. No,
that time.
By in the past, I meant before this particular subtype of this H3N8 reached dogs. Going back 20, 30, 40 years, how many other strains of flu have gone in and out of dogs without anybody noticing?
Yeah.
Because, what we see in humans or in birds or in other animals is that there are circulating strains of influenza, and we just don't notice them because we're not really looking for them. And then something like this happens, you get all these dead dogs, and suddenly somebody notices, hey, these dogs have influenza. So what I was getting at is whether we would see the same types of sort of epidemic and pandemic emergence in dogs, some sort of cycle where the virus changes type from year to year.
Yeah. I don't know. It's possible that it was present before and we just didn't notice it, of course. But now it's spreading, and I think there must have been a proximity of horses and dogs that allowed it to make the species jump, and that may not have existed before, and now it's there, and it's adapted to dogs, and it's spreading through the population.
Wow.
But yeah, we don't have any archival information that would help answer that question. Anyway, I thought that was quite interesting. So there's a vaccine, and I found the product label, which is very cute. It has a couple of little puppies on it. Very nice. So that's canine flu. We have a couple of more stories to come, but first, a word from our sponsors, our good friends at Citrix, the makers of Go to My PC. And many of us have, over the years, come to rely on our office computers and all the data that are on it. And whenever we go away, we always have a mad rush to back up things onto keychains and portable drives to bring with us. And there's really no need to do that because Go to My PC allows you to access your computer from wherever you are. And it's incredibly safe, secure, and simple. All you have to do is go to their website, you click a few buttons, and you open an account. And then from anywhere in the world, you open a browser, you go to gotomypc.com, you type in the code that they've given you, and instantly you see the desktop of your computer. And you can get any of your files, you can even run your programs, run your email, and access your network resources. It's beautifully simple and extremely secure. And you can try Go to My PC free for 30 days. Just go to gotomypc.com/podcast. It's all one word, gotomypc.com/podcast, and you'll have a free 30-day trial. And we thank them for their support. The next story is called Ultrasensitive Detector Promises Improved Treatment of Respiratory Infections. And I think this was a story someone sent. And this addresses the old problem with many acute viral infections, in that there are no antivirals because you usually can't diagnose them quickly enough to make a difference. By the time you have a sample taken and it gets sent to a lab and you have a confirmatory analysis, many of these acute infections are done. And the common cold is a good example. It's over in 3 or 4 days. And it's just too fast. We don't have diagnostics that are quick enough. And I've always said on TWiV that if we had fast diagnostics, then the antivirals would come. So in this paper, it's a collaboration between a virologist and an engineer. They develop a very rapid test, and they basically say it could be put in a pen, in a format like a pen, where you would open the pen and dip it into a sample taken from the nose of a patient, pull it through, and instantly you would have a result. And what they've done, it's a filament-based test. They take a gold filament, and attached to the gold filament is DNA sequence from whatever pathogen that you're interested in. And what you do is you dip the filament into the sample, and if it contains nucleic acid from their particular virus, it will bind to the nucleic acid. Now, the nucleic acid is on this gold in a hairpin format, and it's made in such a way that there's a fluorescent dye on the gold. And when the DNA is in a hairpin, there's no fluorescence. But if your viral sequence hybridizes to it, you then get fluorescence because the hairpin is disrupted and basically there's quenching going on that keeps the fluorescence off. And when the viral sequence hybridizes, you get fluorescence.
Sounds like a good idea.
So basically you dip it in, you draw it through the detector, and then you put it in a fluorescence detector. And then within a few minutes, you could have a diagnosis. So they're developing this for respiratory syncytial virus, which is a serious respiratory pathogen. But of course, the next step would be to go to other viruses. So now they're testing it with patient samples. So if this works, this is brilliant because it's fast. And if it's specific enough, of course, that's the problem.
Right.
Showing specificity, this could be a real development. So
can you wash it off and use it again?
I think not.
That's too bad. That's a big expense for gold.
And I think it's a very thin gold filament. Gold coated. It's gold coated, so it's not solid gold.
Oh, okay.
And it's very small. So this is actually a very interesting story of how virologists work with engineers to develop this, because you need the virologist to tell you what kind of sequences and kind of samples to use, and then the engineers develop the fluorescence probing.
Yeah.
So this was done at Vanderbilt University.
Aha.
So really interesting. Perhaps the detectors of the future, and then this will lead to more antivirals, I'm quite sure. All right, our next story is a rather sad one. It's a case of rabies imported into the United States. And this was actually last year in California. And I think last time we talked about rabies being a problem in China, you may remember. And I thought I would just discuss this briefly to remind everyone that it can still be imported into the U.S., although we don't have much dog rabies in the U.S. We still have wild rabies, and it can be imported. So this was a young man, 16, who came back from Mexico. He lived in Santa Barbara. He didn't feel well, so his family took him to the ER. And they said, yeah, you have pharyngitis, and go home. They sent him home. So three hours later, he came back. He was crying. He wouldn't drink. He wouldn't take fluids. And they said, well, you should see a psychiatrist. And they sent him home. And the next day, he collapsed. And the paramedics came, and he was dead already. And it was subsequently diagnosed as rabies. And what they looked at was said, he didn't like water. He wouldn't take fluids. So that was a key, the hydrophobia.
Right.
And he had come from a place in Mexico where rabies was enzootic. Unfortunately, the initial visit, this wasn't picked up. If they had done a more thorough history, maybe they would have. But this just goes to show how rapidly rabies can be acquired and developed, and it's always fatal unless you intervene with vaccination. So they subsequently did identify rabies in the patient. And some of these physicians who had treated him had to become vaccinated again because they were exposed to his fluids. So it's
apparently it was a rabies variant from free-tailed bats rather than a dog. So
there's a lot of caves down in Mexico.
Yes, in fact, this kid was from Oaxaca,
right,
which is, I believe, known for its caves.
Right.
Yeah, so he probably was in a cave or somewhere where he encountered the bat, and
so you think it was inhalation rabies?
Yeah, they don't actually say. They just say
this is so rapid and fulminating that I would suspect that he inhaled it, and I've heard some stories about that actually.
They don't actually say that. All they know is it was from this particular bat, the free-tailed bat.
Right.
And so the speculation is that that's how he got it, but they don't actually know. And of course, he is no longer available to ask, so it's all speculation.
That's
but it's a very sad case because he was twice in the ER. And, it's a rare disease in the U.S., so it's not the first thing that comes to mind.
So what if they had diagnosed it on the first visit? What do you think they would have done?
Well, Dick, if we had a rapid diagnosis, if you have a pen for every virus, that doesn't work, right?
Right.
Because you can't, let's try the rabies. Yeah. They wouldn't have done that. You need a pen for all viruses, right?
Exactly.
The virus pen.
You'll end up looking like a nerd if you have to take
so you can't do that. But if they had diagnosed it, if someone had been smart and said, oh, this is rabies, they would have immediately started to immunize him.
But he died the next day.
I know. This suggests that he inhaled it.
Yeah, that's right.
And it spread right away through his brain.
And the vaccine doesn't work at that level.
Not to harsh on the emergency room doc. I mean, their time is obviously scarce, but it is certainly, if they'd paid a little more attention to the history and where the person had come from, they might have gone there and gone to that diagnosis a little bit earlier.
Sure.
You're right. But the fortunate part for the victim is that they didn't suffer through the full-blown effects of rabies, which could have lasted several weeks.
Sure.
And it's a horrible way to die, as far as I read.
This is a very typical, though, situation. A rare disease is not readily recognized.
Yeah. There's only one person I know on record that's actually survived
that's right.
rabies.
The Wisconsin person.
She was placed in an ice bath.
Right.
And her core temperature was lowered.
Right. She was placed in a coma, essentially.
She was vaccinated and then placed into a coma. And the virus didn't progress faster than the immune system developed. And eventually the immune system caught up with the virus.
Right.
That was a miracle.
Yeah, that was the Wisconsin protocol, they called it. Yeah. And a few weeks ago, I think you were not here, we had a letter from someone who said there was another case in Brazil, I think, where they had treated them with the same protocol and they had survived.
Amazing.
Okay, then I found an interesting outbreak, or a small outbreak, of Crimean-Congo hemorrhagic fever.
What were you doing there?
And I stumbled upon it. Kazakhstan, southern Kazakhstan.
Yeah.
This was on ProMed Mail.
Yeah.
And this is not a virus we have ever spoken about on TWiV, so here's the time. Turkestan. In the city of Turkestan, a 36-year-old pediatrician died of virus infection, CCHF it's called. Now, she worked at a maternity hospital in the city, and apparently she had treated or delivered a baby of a woman who then subsequently came back after delivering the baby and died of the same infection as well. So, of course, during birth, the pediatrician was exposed to infected fluids, and she was infected. And I think another gynecologist who examined the woman was also infected as well. So we have 2 deaths, and this virus is endemic in this region. So this is a virus that is a member of the Bunyaviridae family.
Isn't it a virus? It's a vector-borne disease. So
what's the vector, Dick? Do you know?
I can see it from here. It's a tick.
It's a tick. That's right. It's a tick-borne.
Is it a hard tick or a soft tick?
It's a hard tick.
Okay.
So what does that mean, Dick?
Well, there are 2 groups of ticks, and one is hard and one is not.
So this is probably irrelevant to the virus.
Yeah.
So it says it's both the reservoir and the vector for the virus.
Right.
Other animals are intermediate hosts: cattle, goats, sheep, and hares.
Yep.
And so this is a enveloped virus of the Bunyaviridae family, which means it's got a segmented RNA genome. It's like flu in pieces, but there are only 3 for this virus. First discovered in 1944, and it's actually a nairovirus because I think that was the initial place where it was, the Nairobi sheep disease. That's when it was first picked up, so it was called nairovirus.
Pretty far away from the Crimea.
Yeah.
Now, this can also be spread through blood-to-blood contact, which I'm guessing it's all these folks.
That's right.
Yeah. So let's see. It gives you a hemorrhagic.
I would assume any of these viruses can be, by the way, Alan.
Sure.
In fact, there was a famous outbreak in Georgia about 4 years ago of an automobile victim whose organs were then transplanted into other people, and they all developed West Nile virus.
So this is a hemorrhagic fever-causing virus. Symptoms: pain, back pain, joint pain, stomach pain, vomiting. You get severe nosebleeds.
Yeah, it's bad
so
news.
that's how you can spread. Of course, if you're giving birth, your fluids are full of the virus, and that's how you can transmit it to others. So it's principally found in Eastern Europe, Soviet Union, former Soviet Union, Mediterranean, Northwest China, Asia, Europe, Southern Europe, Africa, Middle East, Indian subcontinent. But
anywhere but here.
Why isn't it here, Dick? Is it the vector range?
Probably.
So what's the possibility of it adapting to a new vector?
Well, I think if you were to sit down and feed some ticks, they'd probably become infected, just like Lyme disease, for instance. It's found on both continents.
Yeah. Well, as you know, chikungunya virus, only 1 amino acid change allowed it to go to a new mosquito host.
Fantastic.
The Asian tiger mosquito.
Yeah. Albopictus.
Aedes
Aedes
albopictus,
albopictus.
which is in the U.S. So previously that virus was not here.
Neither was the mosquito.
And now the mosquito and the virus. And here,
right.
who knows, this could go into a new tick. Keep an eye on Crimean-Congo hemorrhagic fever
Thank God
virus.
the ticks can't fly.
Although in this case, I'm just reading the ProMed Mail report on this, and it really highlights the importance of universal precautions in medical settings. I mean,
Sure, that's right.
this particular case, the woman who was the mother who was giving birth probably picked it up from a tick, but then this
right.
infection goes on to kill, by my count, a half dozen people at the hospital. The obstetrician delivering the baby died. The surgical team, one of the surgeons and the chief of surgery at the city hospital died.
What about the baby?
The baby died. The mother died. The assistant gynecologist died. I mean, it's just the whole medical staff is getting this from blood to blood contact. Presumably.
Almost sounds like an Ebola outbreak.
Yeah.
Well, that's what happens in medical care facilities. You never know what's coming. You can be in an endemic area, as it is the case, but it can also be travelers coming in, and you wouldn't expect it. Right. So here you could say, well, it's in an endemic region for the virus, but you could be in London and a case of Lassa comes into the ER, and you're not even going to suspect it. So you have to be really well trained.
Well, a case of Lassa did come into Colombia.
Sure.
But good barrier medicine
And
prevents
it went
that.
into London not too long ago.
That's right.
But
right.
you have to have people trained. If someone comes in with a fever, you say, where have you been lately?
Yeah.
And if you remember Harold New, the infectious disease physician here,
very well.
he said the most important thing is a really good history from the
correct.
onset.
That is exactly.
Find out where they've been. He said, tell me where you've been.
It is the most important and the least reliable information in medical diagnosis.
That's right.
Yeah.
And Harold Brown, who was my mentor, used the Shakespearean version of that, Unde Venice.
Okay, Crimean-Congo hemorrhagic fever. Okay, a couple more stories. I think these are all flu stories now. This is an interesting story. The FDA this week approved the seasonal influenza vaccine for the fall for the U.S., and this is based on last year's strains, the H3N2, the H1N1, and the B strain. And it's not clear whether this will be used in the fall. I suspect it will be, but it's not clear that those viruses will be here at the moment. The H3N2 and the old H1N1 are very, very low prevalence. And I read an article that even down in the southern hemisphere, they're quite low. But I thought I would just tell everyone about this because there are actually 6 different manufacturers whose product has been approved for use in the U.S. And they're called Afluria, Fluvaval, Flumist, Fluvirin, Fluzone, and Fluorix by 6 different companies. So actually, when you get an immunization...
And then there's one called Fred.
That's right. That's the one that works, actually.
Yes.
When you go and you get an injection, you don't know which one you're receiving, unless it's Flumist, which is the one unique one which you inhale. This is actually an infectious vaccine. It's an attenuated infectious vaccine that you inhale and replicates in your respiratory tract. And this is really another future vaccine because this completely mimics a natural infection with the virus.
What about the virus-like particle vaccine? Is that an inhaled vaccine also?
No, that would be injected because it doesn't replicate.
Okay.
And so you really, for the inhaled vaccines, they need to replicate so you get good stimulation of immunity at mucosal surfaces. And the virus-like particles won't do that. So all of these are produced in eggs, and they're all inactivated with the exception of Flumist. But of course, this is not what's important. The important question is...
is that going to be like next year's flu?
The swine origin 2009. A lot of these same companies, of course, are working on that vaccine. I understand there are problems because the yield is very low, as we said earlier. It's 30 to 50% of the yield of a typical seasonal strain. So there have been a lot of stories this past week saying there's going to be problems because, most of the flu vaccine is made in Europe. And the few articles I read said, well, these countries want to keep the vaccine for themselves probably. So even if we sign contracts with these companies, we need it for our own people.
It's totally natural to want to do that.
So you can imagine that this is going to be an issue. We're not going to have enough vaccine.
This whole scene reminds me of the joke about the drunk who walks out of the bar and he's down on his hands and knees, and another drunk comes out about an hour later, and this guy's still on his hands and knees, and he says, "Can I help you?" And he says, "Yeah." He says, "I seem to have lost my watch." And the second drunk says, "Well, where'd you lose it?" The drunk points in the other direction. He says, "I lost it over there." He says, "You lost it over there?" He says, "How come you're looking over here?" He says, "The light's better."
Excellent.
And the light happens to be better knowing what's happened rather than what will happen.
Sure.
And so why do we do this all the time, Vince? What's the solution to this problem?
With flu, there's no current solution, but maybe in the future we'll have one universal vaccine, so we don't have to manufacture it each year.
We've talked about that already.
And we have talked about it, and that way, no matter what the strain of the year is, the same vaccine will work.
What about for that M protein you talked about before?
So one possibility is an M protein, or there is a conserved region of the hemagglutinin of the virus, which seems to be promising. And we talked a while ago about one that hits 10 different subtypes, 10 of the 17 HA subtypes, for example. So,
well, and you might
does that also mean it would work well with a pandemic strain as well as the seasonal?
Absolutely.
Yes, it would. That would be the idea, that if this pandemic would be nothing because everyone would have immunity, sure.
Right, and you'd be able to probably vaccinate a lot less often.
Exactly. I mean, even as Tony Fauci said, even if you did it once every 10 years, it would still be an improvement. I think that's coming. Maybe it is 10 years from now, but I'm sure that in 10 years on TWiV 1000, we'll talk about it.
My grandson will be talking about it.
Oh, you're not going to still be with us?
I don't know. 1,000? That's asking a lot, Vince. Come on.
Anyway, so while I was looking at these vaccines, I came across an article just today. FDA says they may fast track the new H1N1 vaccine. So here's what happens. Every year or two, you have a change in the strains in the seasonal flu vaccines. These don't require a new clinical trial of the vaccine. The FDA just requires the companies to make an update on their product license. Sometimes there's safety testing, but typically not much. And the new swine origin virus, of course, requires a new license because it's a new virus. But the FDA is saying, well, we might bypass that and just do the same as for a seasonal flu, because obviously time is of the essence. And I can see that this can be controversial. I'm sure that people are going to pick up on this and say, well, wait a minute, is this safe? And then if there are side effects, which people are already worrying about, then in retrospect, of course, this can be a big issue. Keep an eye on that. I think that's an important issue. That does it for our stories. Anything else anyone wants to bring up before we move on to email? All right, email. Bill writes, I first contacted you back in February with an idea for one of your podcasts. I was very excited when in episode 27, in a call-out that only I recognized, one of your guests referenced the researcher Dr. Lee, whose work formed the basis for the use of reovirus as cancer therapy. Since then, I guess I have to concede that it has been appropriate for H1N1 to appropriate more than its proportionate share of your attention, levity intended. By the way, Dr. Lee, now at Dalhousie, I think, is still working on the virus, and I believe that he has recently published an article showing that reovirus is able to kill cancer stem cells as well as their progeny. It is my hope that you haven't lost sight of this cool story. To make sure, I have copied 2 summaries of recent publications on the approach, each with its list of contributing editors. Perhaps you have the expensive memberships required to access the entire articles. Yes, that's a tip of the hat to your recent open access cast. Still loving your cast whether you get to the reovirus or not. So yes, we will do a show on using viruses for treating cancers. I've been talking with my colleagues about that here, and we're going to do that in the coming months, Bill, we promise. Arson wrote, hey, my name is Arson. I'm an IT specialist from Ukraine. Great podcast. Oh, the Ukraine. That's our first one, I think.
Yeah.
In the last
Not too far away from Crimea, by the way.
That's right. In the last podcast, you were talking about making new viruses by putting genetic material of one virus into the shell of another. I don't understand how that would work, as after the first infection, all new shells would be manufactured by instructions contained in the genetic code. Only the first infection would be somehow different. Could you explain that, please? Absolutely right. I don't recall what we're talking about, but if you put the shell of one virus onto the genetic material of another, yes, you only get one round of you get infection, but then you don't get any more production of that virus because it's a hybrid. And there are some cases where you'd like to do that. For example, if you have a particularly dangerous virus, you could make a virus with its glycoproteins and the genome of another virus. And if you wanted to study virus entry, you could do that, and it wouldn't be so dangerous. So there are some situations where that's advantageous. And we described, I think, a paper some time ago where they used that approach for, now the virus is escaping me, a particularly dangerous one, and they used it to screen for antivirals. Whereas normally it would be very difficult to do that because the virus is dangerous. If you make these mixed particles, you can do it. So that's one thing I think you're thinking about, but maybe the other is that these virus-like particles, maybe you misunderstood, so you can make virus-like particles with no genetic information within the motor wall, and we talked about that in a story just now. So hope that clears it up. Russ writes, I've attached an article that you may find interesting. It is about Americans' view of science. 54% polled understood that antibiotics do not kill virus. That seems a little higher than I might have thought. You're definitely playing a part in changing perceptions. Keep up the good work. Well, I don't know if we've had so much of an effect. Our people who listen to TWiV are really interested.
It's a self-selected group.
It is. And the ones who need to listen probably don't, but we're hoping to catch them over the years. But I think it is always problematic, the public's understanding of science. So he sent me an interesting poll, which was conducted by the Pew Foundation, which turns out does a lot of polling about science understanding in the public. So we'll put a link to that here, and it's quite interesting. But we're trying to educate the public. That's the purpose of this podcast.
Delthia, do you get much feedback from your readers?
Oh, I do, all the time. And they seem to be very curious about, particularly the flu now. And as I spoke to you the other day about West Nile virus, that's another one, and Lyme disease, because this is an endemic area for Lyme. So I do get a lot of feedback from readers about those conditions all the time.
So what proportion of your readership do you think is actually creationist-oriented versus science-based in terms of their belief systems?
Well, it's a pretty sophisticated reading audience here. You know that Cold Spring Harbor Laboratory is here, and you know the people who work there are residents, as well as Brookhaven National Lab.
That's right.
And Stony Brook University. So the residents here are very sophisticated, many of whom are the scientists and researchers who work in the city at Columbia, for example.
Well, that's encouraging then, because that means you can write at a slightly higher level than you ordinarily would be used to.
Yeah, the only difference these days is the fact that the newspaper itself is small. It's literally smaller. We've reduced the physical size of the newspaper, and that's happened throughout the country. Broadsheet newspapers like the New York Times, the Los Angeles Times, have shaved down, by inches, their overall size.
I have an interesting take on that. I subscribe to, I'm an American Chemical Society member, so they give you a choice of journals to subscribe to, and I subscribe to, first of all, everybody gets chemical and engineering news as a throw-in, and then you choose the journal of your choice. So I have one called The Environment and Technology. That's the journal that I get. And the journal, when you open up journals, ordinarily, on the left-hand page is usually the title of the article, and then you've got two columns, and then on the right-hand page, you've got two more columns. Well, I opened up my journal last week, and everything is sideways. They've reduced the font size. They've put a whole 2 pages on 1 page to save paper, and now you have to read it from top to bottom. And I don't know how that's going to go over with the readership, to be honest with you. I mean, I compared it to the previous issue that didn't do it that way, that had the traditional way of presenting journal articles, and they've saved twice as much paper producing it this way. But I don't see it as a readable journal anymore. It's like a record that it's there. I think everybody's going to go onto an online version, right?
Well, they probably want you to.
I'm sure they do. But so how do the newspapers, the newspapers like Newsday, and I know that New York Times is also concerned about the drop in readership, and everybody's now just going to Yahoo News or some other kind of crazy news service that doesn't give you any details. How do you feel about that, actually? Because If you had to write an online version of what you do, is it easier to do it? Do you get to put in more information because you can make connections to places that you couldn't ordinarily do in a printed version, or do you look at it as a concession to the technological electronic age that you would rather not do?
Well, when we do a story, first I have to say it's disheartening to see the demise of the newspaper industry.
I agree.
That's sad. But on the other hand, everything we do for the paper goes online onto newsday.com.
Everything, okay.
Everything. And there are some advantages to online publication because
Sure.
you can write your story a little longer for the
That's
simple
true.
reason you're not restricted
Yeah, that's a good
by
idea.
the physical bounds that you would have in a newspaper.
Right.
You can have more words online than you can in print.
And better resolution pictures too, I bet.
That and actually graphics and
Oh, yeah.
video
Yeah.
video to go along with it.
And video,
So there
right.
so there are a lot of advantages. In fact, they're now encouraging more video along with the story. So rather than going out with just a photographer on the
sure.
story, you go out with a videographer, and then your story has a lot more dimension to it with the videography.
Cool.
Yeah.
So you see there are advantages, Dick.
There are, and plus, if you ever went to a totally online version, all those trees, we could put those back into the
and all
sequestering
the
of carbon.
expenses of printing a paper
and printing and
distributing.
all the waste that goes into it. Yeah,
In time, we shall.
in time. That was not a pun, I hope.
The problem, one of the big problems for the newspapers is that they still have the burden of all of those publishing expenses with the Dead Tree edition, and
That's
they
right.
have this
Well,
digital competition.
The New York Times is way ahead of you guys because they own their own forest. They do. They own a big tract of forest up in Quebec, and they forest 1 quarter at a time, and by the time they get back to the first quarter, it's all grown back.
They replant
They do. They don't have to replant it. Actually, reforests
itself. Yeah.
Yeah, it's an amazing process, actually. They worked that out a long time ago.
So it seems like the online would be easier.
Yeah.
Don't have to worry about the forest.
But that means now everybody has to have a computer. And that
So
means that
it's
you have to
okay.
carry it with you all the time because people love to read newspapers on
I
their
know.
way to and from work and all this other stuff.
We'll get there.
I guess.
Kindles, it's a
I
start.
guess. Kindles, that's a bad...
Well, they get newspapers on the Kindle. So
No, I know, but
Yeah,
the
you
word
do.
Kindle means to burn, I think, doesn't it?
Well, it does
Kindling
if you're
wood.
talking about Orwell books, apparently.
Yeah,
Or Fahrenheit 450. Who wrote that one?
Ray Bradbury.
Ray Bradbury, that's right.
Okay, Peter, P-E-T-R, wrote, "Hello, I'm Peter. I'm a Russian student virologist. I've listened to all of your podcasts. They are really interesting and with a good bit of fun. I was wondering if you could make an episode about prions. They are not viruses, but they make you sick. Actually, I'm working on hepatitis C virus, but I'm very interested in prions. Thank you." Yes, we will do a episode on prions or prions, however you choose to say them.
Right.
Maybe we'll get someone who works on them to join us.
We
That
could
would be
even
great.
do one on Prius, but that would be
Yes,
a hybrid of a different kind.
Oh boy, now we're degenerating here.
Oh, no, we're regenerating.
Martin wrote, "Hi Vincent, I've been enjoying your TWiV podcasts, especially the introductory one you and Dick just did about structure of viruses.
Right.
In the first few minutes, you rhetorically asked, how do we know what the structure is? But then you went on to describe the various viral structures without answering that question.
Oh, dear.
I have heard you on many programs refer to viruses by their DNA or RNA sequences. I thought Dick's idea was great to have a chart with the electron micrograph, the sequence, and a drawing of the virus all together. Is it through the electron microscope that you know what viruses look like? Don't you have to isolate and purify the virus before you take the EM picture? How many viruses have been isolated and purified and then their picture snapped by EM so you know what they look like, and then a sequence done from the purified stuff so you know it all matches up? Thanks for a cool show, Martin." All right. How do you know what the structure is? Well, in the old days, you had EM, and that was about it, electron microscopy. And we got a pretty good idea of what they looked like. And we saw they were enveloped versus non-enveloped in different shapes and sizes.
Some are not symmetrical, though, yes?
Well, the enveloped ones may not be.
Right.
Yes. Some of them are, though, because they have an enveloped around a symmetrical particle.
Yep.
And now more recently, we have structural methods, X-ray crystallography, nuclear magnetic resonance, and cryo-electron microscopy. These are all techniques for looking at the structure at atomic resolution.
And the atomic force microscope.
Atomic force microscopy. So now we can make pictures, and if you've seen these computer-generated images of viruses, you know what I'm talking about. So now we have high-resolution images, and we know what they look like, and more come out every day. So, for example, just this week, the structure of hepatitis E virus was published, and in fact, we're going to talk about that in a couple of weeks. We've never touched on Hep
Very
E.
cool. Oh, and one more thing that would be cool is to have sort of like a Lego set for graduate students. And there's a box of all different kinds of subunits, and you can stick them together and see whether or not you can make new viruses.
Make a new virus,
yeah,
sure.
And it should be out there someplace. It's almost like a periodic table of different families of viruses that should have a similar capsid but slightly different here.
In fact, in our textbook, we have a page you can copy and cut out and fold it to make your own icosahedral virus. That's fun.
There should be a video game like this.
Yes.
Keep it going. Keep it going.
There are actually virus games.
Oh, we had Vince's son on here describing one.
Pandemic, it was called.
That's right.
Okay, what else did he ask? How many viruses have we looked at? Most of representative viruses we've looked at by EM, and we have a lot of structures. Now, you asked how many have been photographed by EM, and then you sequence it. Well, not so many. First of all, you don't have to purify the virus to take its EM. In fact, the SARS virus was identified in patient specimens by
HIV
EM.
too.
HIV, you can see the particles in tissues.
Right.
Doesn't have to be purified.
Exactly.
But very few people have taken the purified material, taken a picture, and then sequenced it, because you don't really need to do that. We have ways of assuring that what we grow up, we keep straight with respect to taking a picture and sequencing. So that has been done less so, but we know, for example, we take a picture of influenza viruses, which pretty much look all similar with certain exceptions, and we can say this is what they look like, and here's the sequence. So that's how we know what the structures are. I know we haven't gone into it in more detail. Maybe we'll have a structural biologist come on and at some point and explain it in more detail. But that can be a bit daunting because they are...
they speak a different language.
They speak a different language, yes.
However, if you're interested in just browsing around structures, there's the Viper database. There may be others. Usually if there's one database, there's at least one more with incompatible information. But I guess Scripps maintains the Virus Particle Explorer. Yeah,
that's a beautiful site. You can go and just look at structures. These are
icosahedral. They are gorgeous.
These are all icosahedral because that's easy for them to do, but they're beautiful. They have pre-made images there. You can just go and browse and you can learn a lot.
I always thought that the T phages were beautiful.
They are beautiful. They're all, Dick, viruses are all beautiful. Even though they do bad things, they are beautiful.
Yeah, I think HIV is kind of
They're an incredible... No, it's beautiful. Beautiful symmetry. All right, that'll do it for email this week, and that brings us to our picks of the week. We'll start with Delthia, who probably has a pick for us.
That was just a wild guess, Vince.
I get to plug my book.
You could plug your book, absolutely.
Well, it's called 100 Questions and Answers About Influenza, and it answers many of the common questions that not scientists would ask, but consumers would ask. For example, it would be, what is the flu? Is it really, is it a separate disease from the common cold? People sometimes don't make the distinction between flus and colds. And as a matter of fact, if you think about how the advertising world has characterized the flu, it gets second billing in cold and flu season. So I make the distinction and tell what types of viruses cause the flu and what types of viruses cause the common cold. It also looks at vaccines and questions about whether it's important to vaccinate children and why, what the important questions are for older people. And then it goes into a very large section on the 1918 flu.
Oh, yeah.
So if you're interested in the 1918 flu, it's all capsulized in the book.
Great.
Delthia, can I ask you whether or not in your book you answer the question, can you have a stomach flu? Can you get the intestinal flu?
I actually do have that, and I say there isn't a stomach flu.
Right.
If you had asked me that before, I would have found the exact page, and I could have read it for you.
Because that's a common term that people use when they think they have something wrong with them and they don't exactly know what it is. I must have had a stomach flu. And, that's just not right, is it?
No, it's not at all. As a matter of fact, this is the question. It's number question 14. It says, so far, there's been no mention of stomach flu or the 24-hour, quote, flu bug.
Right.
Are these infections genuine influenza?
Yeah.
And the answer is, just about everybody at some point has either said or heard someone say, I've got a stomach flu, I've got a 24-hour flu bug. But the terms are so widely used that virtually everybody thinks that there are reams and reams of medical data on how the flu virus causes gastrointestinal problems. And then it goes on to say, forget it because it's just not a real thing.
So you're
Try
dispelling...
norovirus. It's a more likely explanation.
That's right. We could call you the myth buster of virology. Which is a great show on television, by the way. I don't know if you've ever seen it, but it's so much fun to watch because these guys actually take these urban myths or even rural myths and put them to the test.
Is that your pick, Dick?
That's my pick, Mythbusters. I'd pick Mythbusters, sure.
TV show.
As my TV show pick. I could also pick my own book, by the way, on parasitic diseases. I mean, it's only fair. If one guy's going to do it, maybe we should all do it. But and you can do it too, Vince.
Well, I already did it months ago.
How about you, Alan?
Alan hasn't written a book yet, right?
But he
No.
will.
He's going to.
He's going to write the TWiV book, I think.
So, Dick, your pick is what? Either Mythbusters?
Mythbusters, which is a great TV show on the Discovery Channel.
Discovery, okay.
Right. I'll just stop there. That's great.
That's fine. And Alan, what about you?
I have a sort of a technological pick, but it's something that I think would probably be useful for a lot of researchers. It's called TinyChat. It's a website, tinychat.com. And this allows you to get past what, for me at least, is a fairly common problem. People do conference calls, and there's limited productivity to those. But sometimes it's very useful if you could just have a text chat with a group of people, especially if you have a big group that's going to be hard to organize with a conference call, or for some reason it's easier to do by text. Maybe you're going to be discussing things that you want to link to. So any kind of collaboration, it can be a very handy tool to have a chat with your group. Unfortunately, setting up a text-based chat, if you've ever tried to do this, one person is on a Mac and they want to use iChat, somebody else is on a PC and they want to use AOL Instant Messenger, somebody else wants to use IRC, nobody else knows what IRC is. You go to tinychat.com, you click the button on the website, and it creates a chat room on the website. It gives you a link. You send that URL to everybody in your group, and when they go to that web page on any browser, on any computer, they're in the chat room.
Hot damn!
It's extremely useful, and you can keep a log of what happens in it if you choose, or you can just use it as a disposable chat room if you don't particularly want to save the discussion.
That's almost as good as go to my PC, Vince.
Well, I don't know about that. I mean, TinyChat is not sponsoring anything, so
it's different. It's different.
Oh, I was just...
It's a useful, handy tool. They do have video chat options and that sort of thing, but my use for it is for text chats, and it's just very, very convenient to be able to go there and set that up. And
that sounds great.
Do you use that for your work, Alan?
I have, when you have multiple people who need to get together and talk about something.
Yeah.
It's very handy for that, and certainly, if you're in research and you're collaborating with a group of people, somebody's at a conference in a hotel room with poor broadband access, and they could get a text chat going, but their Skype connection would be really bad. This could be a very handy tool, I think.
Great, and you can't beat the price.
You can't beat the price.
All right, Tiny Chat. Well, my pick is a blog, a science blog. It's called Effect Measure. And it is a forum for progressive public health discussion and argument, as well as a source of public health information. And effect measure in epidemiology is the end point of a causal mechanism, if you're wondering what the title is. They have been talking extensively about influenza lately. The last post from today is pediatric complications and deaths with swine flu. Tamiflu resistance, pork producers. Very well done. Good epidemiology. So check it out, Effect Measure. And that will do it for another TWiV. But before we finish, We have a new feature. One of our readers asked that at the end of every episode, we ask a virology question.
Okay.
And we collect the answers in the coming week, and then we read the correct answer on the next show. So I think that's a great idea. This week's question is, what is a viral zoonosis? Give an example. Okay?
Love it.
There's your question. You can send your answers to us by email, twiv@twiv.tv. And, of course, we always like getting your compliments, suggestions, corrections, and questions sent to us. So do that. You can send us an email. You can send us an audio file if you'd like, or you can call us on Skype and leave a voicemail message. And our Skype account is TWiV Podcast. So do get in touch with us. We love hearing from you.
You can even send a check.
Yes.
We will not take cash unless it's in small unmarked bills in a brown paper bag.
Delthia, thank you so much for joining us.
And thank you.
I hope you
Yeah, it
enjoyed
was great.
our
It was great.
discussion.
Oh, I enjoyed it immensely.
Great.
Terrific.
Is there a website where people can find out about you?
I don't have a vanity site, but my stories are always cataloged on newsday.com.
Very good. Thanks so much. Alan, thanks again.
Always a good time.
Alan is at dovedocs.com. You can find him on Twitter at AlanDove. And Dick,
My
thank
pleasure
you.
as usual, Vince.
Keep fishing.
I shall.
Dick has a website that he'd like you to visit, trichinella.org.
Yep.
And verticalfarm.org, of course.
Yeah, but trichinella.org is the science part
It's
of
the
my
science
life,
part.
and I'd love people to go look at it because I've actually made an analogy between trichinella and virus infections. I call trichinella the world's largest virus, but that's in quotes because it behaves a lot like a virus. It's a big intracellular parasite. So
You're using that as a teaching tool.
I am using that as a teaching
Excellent.
tool, of course.
Trichinella.org. And you can follow TWiV on Twitter or on FriendFeed, the same name, PROFVRR. We have a website, twiv.tv, where we have all the show notes and links that we talk about on the show. Thanks, everyone, for listening. Please do subscribe on iTunes if you haven't done so already. Leave some comments there if you wish. We'll be back next week, but in a slightly different form. I will be away this week, so we won't record TWiV next week. But Dick and I have recorded some Basic Virology 101, and I'll put that up next weekend. And we'll be back with the usual suspects in the following week. You've been listening to This Week in Virology, the podcast all about viruses with nucleic acid genomes. Thanks for listening. See you next week. Another TWiV is viral.